So Catriona Jamieson is director of the Stem Cell Research Program at the Moores UCSD Cancer Center. She's assistant professor of medicine. She's trained in medicine, oncology, and actually in stem cells. She actually knew, even when she was a child, that the future was in stem cells. And to continue the analogy, she actually trained with the father of stem cells. She trained with Irv Weissman from Stanford, who really founded this whole field. And she has brought, the basic understanding of stem cells to clinical medicine at a level, unequal in any other field. So she's going to talk to us about a new topic, which is the cancer stem cell, and how understanding that has led to new insights in leukemia. So it's a pleasure to introduce Dr. Jamieson.
Thank you very much, everyone. So today I'm here basically because I want to say thank you. I want to say thank you on behalf of my fellow scientists and physicians, and particularly on behalf of the patients, and I think they're here to say the same. And it's really about unsung heroes. I think in the work that you do, you don't usually get positive feedback. I think there's a lot of public scrutiny and people saying, are you sure you're doing the right thing and spending California taxpayers' money appropriately? And what you've allowed us to do is really focus on the science of human stem cells and human disease, which you'd think would be a well-funded thing to try and get something from the clinic or from the bench to the bedside. It's not well funded. It's not well funded at NIH or even by philanthropic organizations. And then CIRM came along and everything changed. And you allowed us to work in collaboration with biotech companies to make sure that we combined our areas of expertise and got things to our patients as quickly as possible. So we're reminded every day when we walk into our building at the Moores UCSD Cancer Center of what our mission is. We walk by people that have cancer and unfortunately are suffering because of our treatments. Cancer is bad enough, but because of the fallout of the non-specific therapies that we have at our disposal at the moment, people have a lot of tissue injury. And unfortunately, relapse rates are particularly high because frequently we don't get rid of the reservoir of cells that's resistant to chemotherapy and radiotherapy. And those are what we like to think of as cancer stem cells. So, without further ado, I just move on. So cancer stem cells, you've probably heard a lot about them. We tend to think of them as the Achilles heel of cancer. And this was a photograph taken on a trip to Corfu at the Achillean. And what it shows you is even these formidable warriors can have an Achilles heel. They can have a weak point, just like cancer. And you can find that weak point to prevent grief to the people, which is actually what Achilles means in Greek. So we know that cancer is a prominent cause of grief in this country. It's actually superseded heart disease as the #1 cause of death for people under 85. So, of course, I'm biased as a hematologist-oncologist. I see it as the most lethal degenerative disease. So it should be a very strong focus of regenerative medicine. Now, what we're looking for essentially is a needle in a haystack, the cancer stem cell. Now, the cancer stem cell hypothesis was first posited in 1937, so it's been around for a long time. But the tools didn't exist to really examine this hypothesis. And what it suggests is that it's really a small subpopulation within the cancer that has the full capacity to recapitulate all aspects of that cancer. So what it is it's a cell within the malignancy that has been reprogrammed to behave in a much more immature way. So essentially it's adopted or co-opted a lot of the stem cell properties like self-renewal or regenerative capacity, multilineage differentiation capacity, survival and homing capacity. But these aren't well regulated, and that's the problem. So that's what we have to get rid of. We have to not just strike at aberrant differentiation, but also aberrant survival, self-renewal, and homing. So it's a complex thing. So we've been trying to study cancer stem cells, or particularly leukemia stem cells, and how they evolve from the molecular standpoint in the context of chronic myeloproliferative neoplasms, because these are disorders that progress to acute leukemia in a certain percentage of patients, and it really depends on where you started. So if you started with polycythemia vera, that's a disorder where you make too many red cells, your chance of progressing to acute leukemia is low. It's 10 to 15%. If you start with chronic myeloid leukemia, your chance of progressing to acute leukemia is 100% without treatment. So what we'd really like to do is figure out what the difference is. Why do some myeloproliferative neoplasms, where you make too many of one type of cell, progress so infrequently, and others progress frequently. The problem with these myeloproliferative neoplasms, they affect 100,000 people in this country, and they can cause clotting. So the clotting rate is 36-fold higher than the general population. So it's strokes and heart attacks, clotting into the abdomen, clots in the legs, can cause morbidity and mortality, and then transformation to myelofibrosis, where you scar down your marrow and you can't produce bone marrow in your bone marrow anymore. And 3 patients will tell you about what their experience has been with that. It's not a minor disorder. So what we're trying to understand is what are the initiating events that get myeloproliferative neoplasms going, and what molecular mechanisms drive progression to acute leukemia. Now, the reason that stem cells are important is because that's where the events start in these myeloproliferative neoplasms. And as David mentioned, I trained with Irv Weissman, who discovered the human hematopoietic stem cell surface markers, so we can pick these out from the sea of other cells that aren't hematopoietic stem cells. So these hematopoietic stem cells give rise to all the cells in the marrow, and they have to make a decision. What am I going to grow up and become? So in the myeloproliferative neoplasms, they get the wrong cue to go down the myeloid lineage too much. So what we're trying to do is redirect these cells, and that's what this clinical trial is about. Now, in 2005, this is a testament to why science is so important. We were really in the dark, and we didn't know what to go after in these diseases. And we really, as my patients will attest to, had very, very little to offer. And then there were 4 papers published in very close succession showing that there's a mutation in the JAK2 gene. Now, the JAK2 gene, when it was discovered by an Australian, Andrew Wilkes, in 1989, was called just another kinase, because he thought he'd wasted his career trying to clone out this gene that meant nothing. And then it got the more exalted title of Janus kinase, because it can do a couple of different things. But anyway, it turned out to be the most important arbiter for what a hematopoietic stem cell is going to become. And what they found was that it was mutated in 97% of people with polycythemia vera, 50% with essential thrombocythemia, where you make too many platelets, and 50% of patients with myelofibrosis. And so we thought, there is a good target to go after. So we started looking. What we found in polycythemia vera is that they spill hematopoietic stem cells into the peripheral blood, as well as progenitors. And as John Adamson, who's one of our faculty members at UCSD, had suggested in 1976, before the hematopoietic stem cell was identified, was that this polycythemia vera, essential thrombocythemia, and myelofibrosis likely came from a very primitive cell. So he suggested there was a clonal origin. We just happened to be able to sort the cells and show, yes, there is a clonal origin. It goes all the way back to the stem cell, where you can find this JAK2 mutation. And it's transmitted clonally through to the progenitors, common myeloid progenitors, GMP and MEP. So the great thing about being in the cancer center is we have people with disparate abilities and disparate backgrounds. It's small enough that we run into each other in the hall. David Cherish happened to be a co-founder of this company called Targeten, and he said, we've got a JAK2 inhibitor. And I said, no, nobody has a selective JAK2 inhibitor. I've talked to people, it doesn't exist. And he said, no, seriously, there's free wine and free food. So I went up the street, and lo and behold, there was free wine and free food, but also they'd identified this Targeten compound, TG101348, that is an ATP competitive inhibitor. And what we found when we started collaborating was that it blocked that enhanced erythroid differentiation capacity of polycythemia vera stem cells. So when you take somebody's blood or their bone marrow, if you have polycythemia vera, they all want to become red blood cells. They're just getting the wrong cue from that JAK2 mutation. So what this drug was able to do, at least in tissue culture, was revert to normal differentiation. So you got rid of all these red colonies and now started reverting to normal differentiation. And it happened at the level of the hematopoietic stem cells. So they weren't all red colonies and at nanomolar concentrations. So what we wanted to do for a proof of mechanism study is, could we lentivirally overexpress that mutant JAK2 and recapitulate polycythemia vera? And we could, at least again in the tissue culture dish, and this is work done by Charlene Baroga in my lab, together with Edward Kavalerchik, who's one of the CIRM trainees. And basically what they showed was that mutant JAK2 causes these red blood cell colonies to form and just about nothing else, that these red blood cell colonies are exquisitely sensitive to this JAK2 inhibitor. But that's, you can do a lot in a tissue culture dish that actually bears little relevance to patients or to what the outcomes are going to be in vivo. So we developed this bioluminescent imaging model. The reagents for this came from the seed grant where we said we would set up a bioluminescent imaging model of stem cell engraftment. And what we did was we put lentiviral luciferase GFP into hematopoietic stem cells and compared their ability to engraft in this highly immunocompromised mouse with wild-type JAK2 or mutant JAK2, and compared that to polycythemia vera patient cells. So this mouse, I just wanted to make a pitch for immunocompromised mouse models, is highly immunocompromised. It has no TB or NK cells, and so it allows high levels of engraftment, but it's not available to a lot of people. So I actually think that it is important to have these kinds of tools be more readily available for other investigators. We've just been very fortunate to have this. So what we found in this mouse model, and this was published in Cancer Cell, was that the polycythemia vera stem cells were exquisitely sensitive to this drug. So you can see by bioluminescence, the mice treated by oral gavage are starting to lose their engraftment compared to vehicle only. That engraftment is the red blood cell engraftment that's going down, and most importantly, the JAK2 mutant allele frequency is going down. We could see that the cells that were most sensitive to the drug when we genetically engineered this by putting the JAK2 mutant allele into cells were those expressing the mutant JAK2. So that suggests that there is something called oncogene addiction, which lots of people have talked about. And actually Dennis Carson, who's the director of our cancer center, was one of the first physicians and scientists to exploit that because he discovered this drug called cladribine. And that's almost curative therapy for hairy cell leukemia because he figured out that these hairy cell leukemia cells are really dependent on one particular pathway. So we were just following his cue and indeed found that the mutant JAK2 expressing cells were the most sensitive. Now, basically what we're talking about here is reprogramming. The cancer stem cell field has been talking about reprogramming before IPS, induced pluripotent stem cells, came along because that's cancer. You are reprogrammed to behave in a more primitive way, and you have reversion to an embryonic transcriptional program when you start to analyze these cancer stem cell populations. So what we find is we can reprogram these stem cells to take certain cell fate decisions. And this just shows you we can even boost red blood cell engraftment by adding erythropoietin. We can make their spleens very big. We can really force differentiation along one lineage, and that just shows you the quantitative analysis. We're starting to generate myeloproliferative neoplasm leukemia stem cells from embryonic stem cells. And what we're really trying to make is an inexhaustible supply of hematopoietic stem cells in vitro and in vivo. And what we do is we differentiate the embryonic stem cells in vitro on either OP9 or AGM stromal layers. We get them to the CD34 positive stage, and then we transduce them with JAK2 or JAK2 plus beta-catenin. So what this shows you is that the activated beta-catenin, which is an essential regenerative gene, actually enhances CD34 engraftment and colony formation. So what we're trying to provide is for patients who have these disorders like polycythemia vera, essential thrombocythemia, and even myelofibrosis, the next thing in the road. We know the JAK2 inhibitors are going to be important, but what else happens as they progress to acute leukemia, and can we preemptively strike by looking at these regenerative genes that happen to be the hallmark of cancer, like beta-catenin? So the other thing is we have to have a better mechanistic understanding of what these mutant genes do. So what we found, and this is Charlene Baroga again with Edward Kavalerchik, was that GATA1 was highly overexpressed in these JAK2 mutant expressing cells, so they're all wanting to become erythroid, that this JAK2 inhibitor inhibited not just GATA, but also phospho-AKT and STAT5. So it works in two ways. It blocks STAT5 signaling and also GATA phosphorylation. So it's a double whammy, very potent. So that got us pretty excited. We found out that a collaborator at Harvard was doing very similar work with a transgenic mouse model. We decided to combine our efforts, and we submitted our papers together to Cancer Cell. They were published back to back. The clinical trial started in January, so 2008. Was initiated by Targogen at the sites where the research had been done. So it was a group of collaborators. So it was UCSD, Stanford, University of Michigan, MD Anderson, the Mayo Clinic, and Harvard. So we combined our efforts, and we've been accruing patients. We've now finished accrual, even on the extension phase, and I think it's going fairly well. This is just a phase 1, so it's just looking for safety and tolerability. But I really have to say, I don't think that this would have progressed to clinical trials without the infusion of CIRM funding to fund the talented people that actually did the work, without providing the reagent costs, without providing the infrastructure to be able to do this, and without saying, hey, it's okay to work with industry to combine our efforts to do this. So we have 3 people that I think have benefited from this trial who are going to speak, and I think that it's getting to be very interesting. We're going to be looking at ET, PV, and AML for subsequent trials. The one thing that is always at the back of my mind, and was certainly at the back of my mind at midnight last night when I was having a patient transferred to the ICU, is that sometimes people aren't so lucky. They don't, get caught at the stage of disease where it's more readily treatable. They progress to acute leukemia, to blast crisis, and we have to have something more for them. And that's where, unfortunately, these disorders progress. They've acquired a number of mutations, which means that it's unlikely that a single agent is going to work. So we have to come up with a combination strategy, I think, for these people, and for this patient of mine in the ICU who's 40 and has a 3-year-old son, and do it hopefully before they've blasted off. We have to have better diagnostic techniques and combination therapies that are actually tolerated. So this takes us to chronic myeloid leukemia, and I'll go through this fairly quickly because it's so well defined. So chronic myeloid leukemia has been the benchmark for understanding how cancers progress from a chronic disease to acute leukemia. And that's because it was the first cancer to be associated with a characteristic genetic abnormality. Now, it was discovered in 1960 by Nolan Hungerford, and it was called the Philadelphia chromosome. And then Janet Rowley in the 70s showed, hey, it's a fusion between BCR and ABL. And that means you have unbridled Abl tyrosine kinase activity, and these cells start proliferating. And they start differentiating abnormally and surviving too much. And when that happens at the level of the hematopoietic stem cell, it means that the patients get what's called chronic phase CML. So they come in with big spleens. They don't feel that bad. They may feel a little tired. The problem is they've got a ticking time bomb. They have these cells, the hematopoietic stem cell population in CML, that's impervious generally to treatment with Gleevec, which is our #1 molecularly targeted agent for this drug. And it also tends to be resistant to nilotinib and dasatinib, which are 30-fold and 300 times more potent, respectively. So it means that we have to figure out why these people are progressing to blast crisis, what's happened, and can we turn the clock back? Well, this is one of the things that's happened, and we published this in the New England Journal in 2004. And it was a bit surprising. But what we found was that there was activation of this gene called Betacatenin. And this is a central regenerative gene. Our skin, gut, liver, and bone marrow stem cells use this to regenerate themselves. And it should only be on at the level of the stem cell. What we found was, with Betacatenin labeled in red, it was actually on in more committed progenitors. So essentially reprogram these progenitors to behave in an immature way, and these progenitors, called granulocyte macrophage progenitors, couldn't turn it off. So in essence, they behave like leukemia stem cells because they can't turn off that regenerative capacity. They turn on the Wnt reporter gene, make these green colonies, and then they engraft in mice at very high levels, as you can see here by bioluminescence, because they enhance beta-catenin expression. So we just published this in PNAS. This was CIRM funded through our seed grant. We found in these mice, unfortunately, just like my patient, they developed granulocytic sarcomas, and these sarcomas are almost entirely composed of human cells. You can see they're quite bloody. We don't know why that is, because we're putting in this very purified stem cell population, but it seems like they're subverting the microenvironment to have these blood vessels go in and enrich the tumor. These are immature neutrophils, and they're all BCR-ABL positive. What we found, what we said in our seed grant, is that we would compare human embryonic stem cells to CML leukemia stem cells and try and see what made them tick, to see if there were similarities in terms of transcriptional program. And so what we found through sequencing of cDNA was that there was this recurrent misplice, and this is work we did in collaboration with Irv Weissman, in GSK3 beta. Now, GSK3 beta is very important for turning off beta-catenin. If it's not there in a functional way, beta-catenin can't be turned off. So that seemed to happen at the level of the granulocyte macrophage progenitor. And when overexpressed, you can really see that the mice engraft at high levels. When we provide wild-type GSK3 beta, it prevents engraftment and also turns down beta-catenin. So we had this in Nature for a while. The Nature editor said, I think there's more to the story. She was right. There is more to this story. Always listen to your editor, right, Dr. Bloom? So anyway, we went back and we looked and we said, wait a minute, GSK3 beta doesn't just inhibit beta-catenin, it inhibits GLI1, which is the downstream activator of the sonic hedgehog pathway, which is a funny name for a regenerative pathway. But anyway, we started looking at this pathway in CML and found, like others had found before, that it was highly upregulated. But what we found is it was really at the level of the leukemic stem cell, the granulocyte macrophage progenitor population, and we think it's because of this GSK3 beta missplicing. So we published part of these results together with Tanishta Rea in Nature. Again, this is CIRM funded. And what Tanishta showed was that if you knock out smoothened, which is part of the sonic hedgehog pathway, you actually enhance survival of mice that have BCR-ABL positive CML. So it suggested it was really important in mice for blast crisis CML, and it actually decreased the leukemic stem cell population. So this was published April 25th. And what happens is if you overexpress smoothened, and this was Phil Beachy's work, then you actually decrease survival of these mice, and you increase the leukemic stem cells. So what Annalie Abramson in my lab has shown is that, and this is a collaboration with Pfizer, Todd Van Arsdale and Wendy Levin, is that if you inhibit smoothened, which is an essential component of the sonic hedgehog pathway, together with BCR-ABL, you really decrease leukemic stem cell engraftment in the spleen and liver of these transplanted mice. You significantly reduce it in the bone marrow, but it's harder to get rid of in the bone marrow. So what that suggests is that there are protective microenvironments, and that these leukemic stem cells are still responsive to certain microenvironments. So we have to keep that in mind when we're trying to devise our therapies and go after where they like to hang out. So what we're trying to do now is generate these leukemia stem cells. This is Kim Hien Dao. She's one of the CIRM trainees that's actually going to get a faculty position in a month. I'm really sad she's leaving because she's done great work. And what she found was that if you differentiate these human embryonic stem cells on AGM, particularly the urogenital rich, that you get higher levels of CD34 production. And what we're seeing in vivo, just to cut to the chase, is those mice where we overexpress beta-catenin plus BCR-ABL have higher levels of engraftment, at least by bioluminescence. And if you do an shRNA to GSK3 beta, it's even more potent because you're knocking down the capacity to regulate both sonic hedgehog and beta-catenin. So that's where we're at. One last thing, these cells have aberrant differentiation, aberrant self-renewal. They can survive like nobody's business. So that's because they've turned on BCL2. So there are at least 3 things that have got them to be leukemic. So BCL2 is something we can target. This is a collaboration with a company called Coronado Biosciences. They have apogossipol, less toxic derivative of something that was used as a male contraceptive in China for 20 years. But they decided, hey, let's see if it works for cancer. We know the toxicity profile, and it's okay, or at least from some people's standpoint. But anyway, we used it in vivo. We found we could knock down leukemic stem cell engraftment. This is Daniel Goff in my lab, and he's funded through one of our CIRM grants. And what he found was that what this apogossipol does is it changes the leukemic stem cell so it can no longer express the anti-apoptotic isoform of MCL1. So that's a very important finding and something we're going to be preparing for publication. So the upshot of what I've mentioned today is that if we really want to eradicate leukemic stem cells, not nickel and dime people with therapy that's going to have to be given for the rest of their lives and it's extremely expensive, we really have to devise a highly active anti-leukemic stem cell therapy strategy to halt the progression of disease. So basically what we're doing is trying to take a page out of the HIV doctor's handbook and say, you know what, you guys got it right. You were just 10 years ahead of us. And maybe we can start to model ourselves after you and see if the people that we know who have these diseases have these other mutations and say, You have a self-renewal pathway that's activated, but we think we can do something about it. So every one of my patients consents to have blood and marrow taken for tissue banking because we don't know necessarily what we're looking for at the time we're doing a clinical trial, but we sure want to be able to go back. So without further ado, I'd like to introduce you to the patients. I'd also like to thank the people that did the majority of the work in the lab, Annalie Abramson and Ifat Giron and Edward Kavalerchik. Together with Charlene Baroga, they did the work that went to the clinical trial. Kim Hyandao and Jessica Russert are working really hard on the BCR-ABL embryonic stem cell. Daniel Goff, that's the BCL2 work that I showed you, and our collaborators at Stanford, of course, Herb Weissman, and CIRM, most of all, for making this possible. I can't underscore enough how important that was, and that we should also be collaborating with other countries. I've got to put a plug in for Canada, having been from there originally. So I think it's fantastic that CIRM's looking to do this, because I don't think that we'll get there without combining expertise from around the world, basically.